topic for today's public lecture is Unlocking Hearts and Minds. And at first pass that might seem somewhat surprising for our institute, that is the Florey Institute of Neuroscience and Mental Health. But in essence it comes down to whether we take a myopic or a broad view to the science we undertake, and we take a broad view. That spectrum extends from deep interrogation of fundamental research, such as understanding how our DNA works, the integrity of the DNA, right through to field research, studying how environmental challenges, such as microplastics, influence mental health. And that spectrum of interest is relevant to today's lecture in terms of our commitment to understanding the body-brain interface. And there's no better example of that than the recognition that the disruption of the cerebral circulation can have profound effects upon the integrity of brain function, both in the short and long term, and we're extending well beyond the so-called classical stroke. And a particular note is that we as doctors disrupt that circulation every day in cardiopulmonary bypass surgery. So how do we make that intervention safer? To do so requires a deep understanding of the mechanisms potentially at play to disrupt cerebral circulation. And those factors include ischemia, or lack of oxygen, depletion of micronutrients such as zinc, and also neuroinflammation. And these are factors that might also influence the health of other vital organs, such as the kidneys. Now, we are fortunate to have at the Florey a team working under the umbrella of the systems neuroscience theme to answer these questions via cutting-edge, large animal research. But research of this nature also requires deep collaborations with others, including intensivists, anaesthetists, cardiothoracic surgeons, cardiologists, and renal physicians. And this is important, impactful work which needs to be celebrated. So with that in mind, we've assembled what I'll term the A-team for today's lecture to share with us their vision of a brighter future. And that A-team consists of 2 of our own and 3 of our collaborators. That is firstly Associate Professor Yugeesh Lankadeva, who leads the systems neuroscience theme, Dr. Lindsea Booth, who's the head of the neurocardiovascular physiology group here at the Florey, Associate Professor Lachlan Miles, who's a staff specialist and head of research in the Department of Anaesthesia at Austin Health, Dr. Emily C., who's an intensivist and nephrologist at the Royal Melbourne Hospital, and last but not least, Professor Rinaldo Bellomo, who's Director of Intensive Care Research at the Austin Hospital, and interestingly, actually the most published intensive care investigator in the world. No mean feat. So without further ado, I'd like to hand over to Yugeesh to formally open proceedings today. Thank you, Yugeesh.
Thank you very much, Trevor, for that very kind introduction, and thank you everybody for being here today. We are very excited to launch the first Florey public lecture for 2024 that's titled Unlocking Hearts and Minds. My name is Associate Professor Yugeesh Lankadeva. I've got the honor and privilege of leading the systems neuroscience team here at this prestigious medical research institute. So I will launch straight into it by talking about the clinical problem that we are trying to address, which is a striking statistic that's put up by the National Heart Foundation of Australia, that every day over 150 Australians are hospitalized due to a heart attack. Now, not everybody survives a heart attack, and if you're lucky enough to survive a heart attack, unfortunately, this can be a common index event that leads to development of heart failure in these survivors, and these patients will require heart surgery if their heart deteriorates below a certain level, to undergo heart surgery requiring cardiopulmonary bypass to improve their heart health. Now, I have a personal relationship with this patient scenario because this is my brother-in-law, Sasiwanan, who is 1 year older than me. He's a father of 3 small, beautiful girls. And unfortunately, he had a heart attack 3 years ago. He was lucky to survive this, but his heart went into failure within the last 3 years. Now his heart function has declined to under 10%. He's currently on a heart transplant waiting list for a donor heart to improve his heart health outcomes. However, I want to emphasize that having a heart attack and developing heart failure not just affects your heart health, it affects other vital organs like the brain and the kidneys. And it's really difficult to understand what is causing injury to these vital organs in this vulnerable population of patients. This is why we need to come up with a way to develop preclinical models at medical research institutes at the Florey to understand how injury is done to these organs so we can understand it and develop more targeted diagnostics and therapeutics to help patients going through this complex patient journey that devastates not just their lives, but also the lives of their family and loved ones. So why the Florey? The Florey has always had a rich history of being a pioneer of techniques, not just to assess brain health, but also to assess other vital organs like the heart and the kidneys. And Professor Clive May, who's in the audience today, has been a true inventor and pioneer of these techniques that allows us to assess brain, heart, kidney health in real time as in health and disease. And this really forms the ethos of the systems neuroscience theme that I've got the privilege of leading now. And over the last 25 years, Clive's developed these techniques and passed this knowledge down to the likes of myself and my colleague, Dr. Lindsea Booth, you'll hear from later on. So we really benefited from this scientific know-how. However, having the scientific know-how is not enough. This is a very complex patient journey, and we really require the input of multiple people, including clinicians that are at the coalface treating these patients going through this. So with that in mind, we brought together a very unique team of Florey scientists and clinicians involved in every aspect of the patient journey, from having a heart attack, developing heart failure, requiring heart surgery, and out again with complications arising with their brain and kidneys. We included cardiologists, cardiac anesthetists, clinical perfusionists, intensivists, nephrologists, and psychiatrists from Royal Melbourne Hospital, Austin Health, and Townsville Hospital. And most of these clinical colleagues are sitting in the audience, so I hope you take the opportunity after the lecture to interact with the entire team. It required this unique group of people to come together. We also brought together industry partners, and I want to particularly mention Cell Saving and Perfusion Services, the managing director of this company, Mr. Bruno Marino. At the inception of this project 7 years ago, we didn't have enough funding to get this up and running. Bruno was very kind enough to generously donate not one, but 2 heart-lung machines to us, to the Florey, which is worth hundreds and thousands of dollars, as well as give up his time pro bono by coming to the Florey every week over the last 7 years that allowed us to develop this world first preclinical model. So I want to really thank Bruno, who will be in the audience here today as well. In addition to this, we also brought together other industry partners like GeneWorks that's going to help us develop neurological blood-based diagnostic markers of brain injury that you'll hear from later on. And we also brought in a company from the United Kingdom, Oxford Optronics, that allowed us to design custom-built probes we can insert into the brain and kidneys to understand injury mechanisms and then develop a diagnostic for kidney injury arising from these conditions, which you'll hear from later on. So what happens when this amazing group of scientists, clinicians, and industry partners come together? What we have been able to do over the last 7 years is really co-design a world-first large animal model platform to simulate the entire patient journey, from having a heart attack to developing heart failure to going through heart surgery and out again in the intensive care unit, with the unique ability to continuously monitor brain, heart, and kidney health so we can understand disease mechanisms, so we can develop new targeted diagnostics and therapeutics. And us as a team, we have a joint vision to improve brain, heart, and kidney health outcomes to these vulnerable patients that develop heart failure and then require heart surgery. So without further ado, I would like to now introduce my close colleague and collaborator, Associate Professor Lachlan Miles, who's head of anesthesia research at Austin Health, to talk about brain injury after cardiac surgery, the double-edged sword. So thank you, Lachlan.
Anyone who read the New York Times on Sunday, May the 10th, 1953, could have been forgiven for thinking it was a slow news day. Looking at that same edition now, page 1 above the fold tells us that negotiations to end the Korean War were ongoing, that educational shortages in science, teaching, engineering and medicine were an emerging problem, and that President Eisenhower had gone fishing with his brother. However, if you had persisted, and if you had made it all the way to page 10, you would have found a short article that reports an advance made possible by Dr. John H. Gibbon of Philadelphia, Pennsylvania. A surgical procedure performed in an 18-year-old woman to enable a hole between the top 2 chambers of her heart to be closed. You might not realise this at the time, but this short article, all 3 paragraphs long, heralded the biggest advance in surgical science since the discovery of anaesthesia in 1846, 97 years beforehand and 300 miles away in Boston. What Gibbon had achieved represented the culmination of 68 years of work by various individuals. He had, in an anaesthetised person, successfully diverted blood away from the heart and the lungs to enable a surgical procedure to be performed on a still and bloodless organ. Now, to fully grasp the nature of this advance requires a brief lesson in simple physiology. The whole function of the circulation is to ensure a continuous supply of oxygen to the organs and facilitate the removal of carbon dioxide. Blood, low in oxygen and high in carbon dioxide, returns to the right-hand side of the heart through the veins. From there, it travels to the lungs, it's pumped there by the right side of the heart, where oxygen is added, carbon dioxide removed, and returns to the left-hand side of the heart, and from there is pumped out to the rest of the body. From this simple explanation, you might divine that such a system requires two things. It requires a pump, the heart, to keep the blood in constant motion, and it requires an oxygenator, or the lungs, required to remove carbon dioxide and add oxygen. This simple concept is what Gibbon managed to operationalise, creating the first cardiopulmonary bypass circuit, which some of you may know by the more evocative term heart-lung machine. And whilst there have been considerable advances in perfusion technology in the intervening 70 years, the basic principle remains the same. Blood is drained from the right-hand side of the heart, passed through a pump, oxygenated, and returned to the body, again and again and again. John Gibbon had made operating on the heart safe, and in doing so birthed a new surgical specialty. Cardiac surgery is no longer the bloody, groping business of the early 20th century with an unacceptably high mortality, but rather safe and routine surgery. Cardiac surgical procedures are performed on over 2 million people per year, which represents a fraction of the number who actually need access to it. Complex congenital heart defects that might have consigned children to an early and painful death can now be corrected with a mortality rate as low as 5%. Lower risk procedures or conditions in adults for conditions like coronary artery disease or valvular heart disease that result inevitably in death if not corrected carry a mortality rate now as low as 2%. Now, further decreases in the rate of death for the conditions that I've just described to you are probably marginal, and so necessarily the focus of our research efforts has had to change. It is no longer enough for an individual to simply survive cardiac surgery, because you must recover well, because if you don't do so, if you have no quality of life, one might argue there was no point in undergoing the procedure in the first place. Now, it's at this point that the unspoken question of this lecture heaves into view. What am I, a cardiac anaesthetist whose day-to-day clinical practice should concern the function of the heart and the lungs, doing presenting a public lecture at a brain institute? It's because the most common complications of heart surgery are not cardiological, related to the heart, or pulmonological, related to the brain, related to the lungs. They are neurological, related to the brain. And regrettably, brain injury is a common occurrence after cardiac surgery. By this, I don't necessarily mean a stroke, although this certainly does affect 1% of patients after cardiac surgery. Instead, I refer to delirium, a neurological complication of cardiac surgery which is seen in up to 40% of patients after their operation. Now, most people recognise the term delirium. Indeed, you yourselves or your loved ones may have a personal experience of it. The most stereotypical manifestation is the profoundly disturbed patient, completely disoriented, shackled to the bed and chemically restrained because of risk to themselves and risk to the staff. This hyperactive delirium, the stereotypical delirium, though, only accounts for about 10% of cases. But when we test for delirium properly, when we actually go looking for it, the actual incidence is much, much higher. You see, delirium does not need to have this overt behavioural component associated with it, per se. To make the diagnosis, you need three things. One, you need to make sure that you have a reduced ability to direct, focus, shift or sustain attention or awareness. You need to have an additional disturbance in memory, orientation, language or perception, and you need for it to develop in a short space of time. Now, previously, we rather naively thought that post-operative delirium is a worrisome but ultimately transient benign condition. Once it's resolved, we thought the patient will return to normal and live a full and happy life. We now know that this is not the case. Patients who develop a delirium, whether it be obvious immediately or not after their operation, have a higher risk of death within 1 year of surgery, often have problems with their cognition lasting weeks to months and have a higher risk of developing dementia in the 10 years after surgery. What is just as concerning as these worrying figures is the real-life impact of post-operative delirium. These usually fall into three categories. Firstly, the multifaceted nature of delirium. Here, a patient named Conrad describes the disturbing experience of believing it to be raining inside his hospital room after heart surgery. Secondly, a patient named Jeff talks about the functional decline that followed a post-operative delirium after his coronary artery bypass surgery, not being able to remember the names of friends that he had known for nearly his entire life. Finally, there are the emotional consequences of what is in effect a chronic illness described by Pete, who was left with feelings of guilt and shame after he experienced post-operative delirium and persistent cognitive dysfunction after his heart surgery. And so, having described this wicked problem, my colleagues and I found ourselves looking to the future. We recognise that successful surgery is no longer just about survival, but a return to previous function and previous independence, and that the most common barrier after heart surgery preventing this from happening is delirium and delayed cognitive recovery. As an older and wiser patient remarked to me earlier in my career after a complicated recovery after cardiac surgery, I thought that the worst thing that could happen to me was death, and I was wrong. These complications are common, and their consequences are of great importance to we as clinicians and to you as the public. However, progress in this area has been cruelled by our lack of knowledge. Despite 70 years of literal blood, sweat and tears, we do not know for sure what the cause of delirium after cardiac surgery is, making it difficult to predict, to treat and to prevent. And so, having come up with very little to show for our own effort, we are here to ask, can the Florey help? And with that, I'd like to invite my colleague, Dr. Lindsea Booth, to come up and discuss the most recent findings. Thank you.
Thank you, Lachlan. So before I talk to you about what drives delirium after cardiac surgery, I'm just going to ask another question. Why is it important to know? In order to identify effective treatments, we must understand the mechanisms driving the changes within the brain after heart surgery. And how do we find out? Well, Yugeesh has already introduced you to the model that we've come up with in the lab, which tracks a patient journey from a heart attack all the way through heart surgery and recovery in intensive care. And to run this, we need an inspired team of scientists, clinicians, research staff and students. And before I show you any data, I just want to acknowledge all the people in these photos, because without them, this data would not exist. So what drives delirium after heart surgery? Well, there are a number of factors that have been proposed to contribute to delirium, and I'm just going to share a few of them with you now. So one theory is that there's low oxygen in the brain during heart surgery, which would cause damage to the brain tissue and therefore delirium. Another is low blood flow to the brain during surgery. An important factor is pre-existing heart disease, such as heart failure, and this could predispose a patient to delirium. And also breakdown of the barrier around the brain, also known as the blood-brain barrier. This usually protects the brain from circulating factors in the blood, and it's known that this can break down in heart surgery. So this, as well as all of the other factors I've shown here, can all cause inflammation of the brain, which can lead to delirium. And that's what I'm going to focus on today. And just before I move on, using the model that we've previously described, we've actually recently shown that by measuring oxygen within the brain during heart surgery, we've shown that there's no low oxygen in the brain, and also that there is no low blood flow to the brain using current clinical practices. So what about inflammation in the brain? Well, I'm going to introduce you to one of my favorite types of brain cell here. This is a microglia. So you can see it's got this small cell body here and these long branches out into the brain tissue, and that's how it monitors brain tissue health. They are the resident immune cells within the brain, and in response to an insult or an injury, they become inflamed or activated. Here is an activated microglia with a larger cell body and these shorter processes, shorter and fatter processes. Here is a highly activated microglia, so it's got that large cell body and very small, thick processes. And microglia are along this activation continuum. So this is the healthy microglia in normal tissue, and this is in an activated state. So using our model, we're able to tag these microglia with fluorescent markers so we can get an indication of how activated they are in and after heart surgery. And this is one of the fluorescently tagged microglia images taken here. So you can see here these small cell bodies and the long branches in green. So this is from healthy brain tissue from the frontal lobe, which is at the front of the brain and is important in executive functioning. These are all ramified or resting microglia. So the image you see here is actually a reconstruction of multiple images taken with using a microscope that we reconstructed to form a 3D model of microglia. So how do we measure activation in these microglia? Well, we take the images like the one I showed you before, and we use artificial intelligence to help us to identify individual microglia, which we then take and make into the skeleton structure. And this is done in collaboration with Song Yao at the University of Melbourne. So using the skeletonized structure here, we can measure a number of variables. One, we look at how far apart are these microglia. So for every image that we take, how many microglia are we seeing in there? We also look at how many branches does each microglia have. As the image I showed you before, the more branches they have, the less activated they are. We also look at how long the branches are. So if they're more activated, they've got very short branches. And we look at how big the cell bodies are. So using these 4 measures, we can get a level of activation in each image that we take. So what do they look like in heart surgery? So this is the normal healthy control that I showed you previously. And this is an image taken at the end of heart surgery, so with no pre-existing heart disease. And you can see the microglia here. Some of the cell bodies are slightly bigger, and the processes are slightly thicker and slightly shorter. And this is work done by Anton, Victoria, and Garvey in the lab. And we've recently published these findings in 2 papers showing that there is inflammation in the brain at the end of heart surgery in our model, and especially in the frontal lobe, which I was talking about before. So where to next for us? Well, we're really interested in what happens with pre-existing heart disease. Yugeesh spoke earlier about our model of heart failure. So is there more activation of the microglia in heart failure? Also, how long are these microglia activated for? The image I showed you is from the end of heart surgery, and we're now doing 2 groups, looking at what happens in the 48 hours recovery after heart surgery, and also 30 days, and this is really exciting work. So I'm now just going to show you a little bit of preliminary data from our model of heart failure, looking at 48 hours after the end of heart surgery. So these are the 2 images I showed you before. We've got a healthy control, and this is at the end of heart surgery with no pre-existing heart disease. And this is an image taken after 48 hours after heart surgery with pre-existing heart disease. And this is just preliminary data, but you can see that these are highly activated microglia. They've got large cell bodies and very short fat processes. And this makes up part of Anton's PhD, and it's going to be really exciting to see where this leads. So how can we use this inflammation that we're seeing in the brain to change clinical practice? Well, obviously we can't look at microglia in patients. But what we can do is we can look at blood-based biomarkers of brain inflammation to get an idea of what the level of inflammation in patients is. And one of the molecules we've been looking at is neurofilament light chain. So this is released when brain cells are damaged, and it's been validated across multiple neurological disorders. So this can be measured using single molecule array or Simoa technology, and this is work we're doing in collaboration with Scott Ayton and Emilio Verdin at the Florey Institute in collaboration with GeneWorks. So what do we find when we measure neurofilament light chain? So in our model of heart surgery, I'm just going to show you 2 graphs here. We've got plasma neurofilament light here on the y-axis, and on the x-axis we've got before, 12, 24, 36, and 48 hours after heart surgery. And I think you can see here there is a clear increase in the plasma neurofilament light after heart surgery. How does this relate to the patient, to patients? Well, in collaboration with Lachlan, who spoke before, we've just done a small study looking at patients undergoing heart surgery and measuring plasma neurofilament light here. And this is before 1, 2, 3, and 4 days after heart surgery. And I think you can see here that there's a clear increase in plasma neurofilament light here as well. And so this is a really exciting area of work that we're moving into. But I will acknowledge, though, that the brain is not the only organ affected by heart surgery, and I would now like to introduce you to Emily C., who will be talking to you about kidney injury.
Thanks very much. So the brain is certainly not the only organ that's affected during cardiac surgery. In fact, kidney damage is one of the most common complications in this high-risk group. Acute kidney injury, as the disease is called, is a term that describes an abrupt reduction in kidney function that occurs over hours to days. So in patients undergoing cardiac surgery, as many as 30% of patients will develop acute kidney injury as part of their surgical course. In the short term and in the long term, acute kidney injury is associated with numerous complications for patients. It's associated with an increased risk of death, a higher chance of requiring burdensome life-supporting therapies, prolonged stays in the intensive care unit and in the hospital and early readmission to hospital. And in the long term, it's associated with permanent, irreversible kidney damage and potentially the need for long-term dialysis. It's, as you can imagine, with all of those, the nature of those complications, it's a disease that's very burdensome to both patients but also to health services in terms of resource and cost. There are multiple different reasons why patients undergoing cardiac surgery may develop acute kidney injury, and in many patients the process is likely multifactorial. We currently have no diagnostic tests or tools to help us differentiate between the cause, and as such we've got no effective targeted therapies. If we look at some of the more common causes or contributors to injury in patients having cardiac surgery, firstly, these patients have a reduction in the oxygen delivery to their kidneys due to changes in the blood vessel tone that occur with the cardiopulmonary bypass that Lachlan detailed. Secondly, these patients are in a state of high inflammation following exposure of their blood to the plastic of the circuit, but also to air and fluid. Next, they have decreased kidney blood flow. By nature of initiating the cardiopulmonary bypass, there's clamping of the aorta, followed by unclamping, which obviously alters blood flow to the kidney since the aorta is its main source. And finally, these patients are commonly exposed to toxic medications that may impact their kidney function, including antibiotics and iodinated dyes that we use in scans like CT scans. Importantly, the relationship between the kidney and the heart is bidirectional. So chronic kidney disease is one of the strongest risk factors for the development of cardiovascular disease, and these are the types of patients who go on to then require cardiac surgery. As such, it's very common that patients having cardiac surgery do have underlying kidney disease, and this puts them at an even higher risk of developing acute kidney injury. Moreover, they are at a higher risk of developing the most severe type of kidney injury because they've got limited reserve in their kidneys due to years of chronic scarring. Importantly, for patients who develop severe acute kidney injury and require initiation of dialysis to support or to remove toxins and fluid, this may have important implications in the early postoperative phase for how their heart is functioning. So it's certainly there's multiple links between these two organs, and the kidney is very much like the brain at the heart of complications for patients undergoing heart surgery. The major barrier to having effective diagnostic tools and also therapies, the main barrier to optimal care is our lack of diagnostic tools and therapies, and in particular our current inability to understand which mechanisms are the predominant contributors to injury in an individual patient, because it's likely that the dominant mechanism varies between patients and potentially even within patients along the time course. And in the absence of being able to understand what's driving injury, we have no effective therapies for this complication. As such, its management is largely supportive. So that means we try and prevent any further injury. We try and treat complications as they come up, but there's no magic pill or magic treatment that can help patients with acute kidney injury. The most severe form of acute kidney injury results, as I mentioned, in accumulation of toxins and fluid in the blood, and this requires urgent treatment with dialysis or renal replacement therapy to remove those toxins and excess fluid. And this has important implications both for the organ function of the kidney and also the heart, but also of the patient's rehabilitation, moving out of the intensive care and eventually moving out of hospital. Renal replacement therapy also increases the stress on the circulation, so it can make patients' blood pressure very low and can mean that they need to get readmitted to ICU after being discharged. So it creates a lot of problems for their flow through their hospital journey. As such, acute kidney injury represents a major unmet need, both from a diagnostic perspective in both identifying it early but also differentiating between key mechanisms, but also a therapeutic challenge in terms of the absence of any effective therapies. And this is something that we're very much hoping that the Florey will be able to help with the work that's going on through our collaborations. And I'll now invite Associate Professor Yugeesh Lankadeva to tell us about some of those programs that are running.
Thank you, Emily. And look, as Emily mentioned, not only does patients with pre-existing chronic kidney disease predisposed to acute kidney injury, but sometimes acute kidney injury can develop as a consequence of heart surgery, even in the absence of pre-existing kidney disease. Concerningly, patients that survive short or mild episodes of acute kidney injury, as Emily says, are predisposed to a greater risk of developing chronic kidney disease and end-stage renal disease that can substantially diminish their quality of life. So with our clinical colleagues, we seek to see whether we can help at the Florey using our animal model. So as I mentioned, we have developed a globally unique surgical technique to instrument the kidney in a way that nobody else can. We partnered with Oxford Optronics, our UK industry collaborator, to develop custom-built fiber optic probes that can be inserted to the outer region of the kidney, which is called the kidney cortex, and the inner region of the kidney, which is called the kidney medulla, to assess blood perfusion and oxygenation in these two very important functional regions of the kidney. We can also instrument the main kidney artery that takes oxygenated blood to the kidney to assess how much blood is going to the kidney. And then we can also cannulate the renal vein that takes deoxygenated blood from the kidney back to the heart and the lungs to be oxygenated. So this allows us to get a really nice understanding of both the macro and micro circulation of the kidney in the conscious state, and then track it when the patient goes from a conscious state to general anesthesia and then on to heart surgery using the heart-lung machine or cardiopulmonary bypass. So that's what we did using our large animal model of heart surgery. You can see in the black bar, that's the conscious state of kidney blood flow. Just going through general anesthesia alone causes a significant reduction in kidney blood flow. So clinical perception was right here. However, under anesthesia, because the patient's been machine ventilated with high supplemental oxygen, the inner region of the kidney or the kidney medulla seems to be preserved of oxygen in this state. I'm going to concentrate more on the inner region of the kidney because this is an area of the kidney that is very susceptible to injury during disease conditions. Now, you can see that when they transition from general anesthesia to cardiopulmonary bypass, it's almost like a second hit to this kidney. Kidney blood flow drops even further. And now the kidney medulla is significantly deprived of oxygen. And this we have shown to be is a key mechanism leading to the functional deficits that manifest as acute kidney injury, not just in heart surgery, but also with other etiologies like sepsis, which is severe infections. Now this is great. We can look at this in mechanistic details in preclinical models, but we can't go around sticking probes in people's kidneys. So the question we asked ourselves with our clinical colleagues is, can we develop a non-invasive biomarker to detect kidney medullary hypoxia in patients undergoing heart surgery? Now back to the drawing board. Just as Lindsay can fluorescently tag the brain, we can also fluorescently tag the kidney. And now this very elegant image here shows the collecting ducts in the kidneys, labeled in blue. Now, collecting ducts is when the blood is filtered in the kidneys, going through the reabsorptive process. That's where it's collected before it gets sent down the ureter into our bladder. Now, that's in blue, and the kidney medullary blood supply is labeled in red. You can appreciate that these are positioned very close and symmetrically around each other. Due to this close anatomical relationship, we hypothesized that the oxygen levels in the kidney medulla would equilibrate with the oxygen levels in the bladder urine. Why is that important? It's because, as I mentioned, once it's in the collecting ducts, it gets passed down the ureter into our bladder. Now we got together with our industry partner, again, Oxford Optronics, and developed a large area oxygen sensing probe that can be tunneled through a standard Foley bladder catheter, so it sits at the tip of the catheter in direct contact with the patient's urine. And we said, if this gives us a signal of reductions in oxygen levels, this is going to be a non-invasive biomarker of kidney injury, risk of kidney injury in patients going through heart surgery. Now, I know what most of you are thinking. Insertion of a bladder catheter is hardly non-invasive. Nobody wants a bladder catheter inserted into them. However, this is the beauty of clinicians and scientists working together. In this clinical scenario, the placement of a catheter in the bladder is routine clinical practice in patients undergoing heart surgery. So inserting a probe to the tip of the bladder catheter is non-invasive in this clinical indication. Now, we went back to our animal model and looked at the relationship between kidney medullary oxygen levels and bladder urinary oxygen levels over the development of acute kidney injury over 24 hours. And you can appreciate that the reductions and the profile of kidney medullary oxygen levels is very closely reflected by looking at the oxygen levels in the bladder urine over 24 hours of kidney functional defects in this condition. Now we have been able to translate this with our clinical colleagues, and Associate Professor Andrew Cochrane, who's standing at the back, led this study in a cohort of 80 patients going through cardiac surgery at Monash Health. And what they were able to show is that in over 80 patients, 26 of these patients developed acute kidney injury in the intensive care. And when they looked at bladder urinary oxygen levels, there were reductions in bladder urinary oxygen levels through all phases of surgery, as you can see, in both groups. However, the patients that developed acute kidney injury had larger reductions in bladder urinary oxygen levels. That signals that the kidney medulla is hypoxic. So this it seems to be a good marker of risk of acute kidney injury even before it develops. So early preventive strategies can be employed. I'm very happy to say that our Florey biomarker has now been adopted to clinical trials across 4 continents, in North America, Europe, Asia, and Australia, in patients undergoing heart surgery to detect risk of acute kidney injury. Now, a diagnostic is great, as long as we have a way to prevent it. As Emily said, we only have conservative methods, which means we are refraining from nephrotoxic drugs and hoping the kidney recovers in the patient. The question we asked ourselves is, can we use the heart-lung machine to modify perfusion conditions to optimize oxygen delivery to the kidneys during heart surgery? Now, we tried various maneuvers. We tried inducing a pulsatility, i.e. simulating a beating heart with a pulse through the heart-lung machine. Didn't do anything. We tried giving more donor blood to increase the oxygen carrying capacity of the circulation. It didn't seem to do anything. But very successfully, what we found was that if we were to increase target pump flow, i.e. simulating the heart beating harder by 10%, and increasing target blood pressure by 15%, which is a very clinically feasible range, if you do these 2 maneuvers simultaneously, we can return oxygen levels in the inner region of the kidney towards healthy, conscious levels. And following this, with our clinical colleagues again, with Associate Professor Andrew Cochrane, he led a clinical trial at Monash Health, and then Professor Rinaldo Bellomo ran another clinical trial at Austin Health to show that this low-cost intervention was safe, it was feasible, and we used bladder urinary oxygen tension to show that that gives us a very good indication of improvements in kidney oxygen delivery as a surrogate measure. So bladder urinary oxygenation is not just a measure of risk, but it can also be used as a guide for clinicians to optimize perfusion conditions to reduce the development of acute kidney injury. And currently there are larger clinical trials that are being planned to test this low-cost intervention to reduce the incidence of acute kidney injury arising from heart surgery. So with that, I would like to invite Professor Rinaldo Bellomo, who is Director of Intensive Care, to close this session by talking about the power of translation.
Thank you, Yugeesh. Before I start, I must confess that every now and then I get despondent about this beloved country of ours. And I see the younger generations pursuing TikTok and watching maths instead of pursuing knowledge. But not tonight. Not tonight. After seeing these young, wonderful investigators speak to us, there is hope for us yet. So you've seen the challenges that we're dealing with. They're very hard. There are some key concepts that we apply to try to understand the universe in which we work, and the human body has an extraordinary level of complexity, as complex as the entire universe. And the challenges are many, and they're really difficult to tackle. And the only way that we can move forward is by creating a model in animals and in large animals where we can monitor the things that we need to understand in order to move to better patient care. And if you think like this and you ask yourself, where do we start? The starting point must always be the pursuit or protection of the vital organs. And you've heard about the brain and you've heard about the kidneys. And you can use small animal models to try and understand this, but at some point it doesn't really work. At some point you need large animal models where you can replicate what actually happens to human beings in a complete and true way and make the things that you do relevant and deep. And as you've heard tonight, the Florey has done this for the first time in the world. There's no other laboratory system, large animal experimentation system, anywhere on this planet at this time that can do this. They brought together clinicians, perfusionists, anesthetists, intensivists, cardiac surgeons to create a team, nephrologists, that can do this. And we've created an animal model that replicates what happens to human beings. And that's incredibly important. And now you've seen what we can do. It's extraordinary. We can understand things that we cannot understand in human beings. We can understand what happens to blood flow, what happens to oxygen, to the brain, the small blood vessels, the kidney, the brain, the cells in the brain, how they puff themselves up and get inflamed. We can see whether they return to normal, how they do so. We can see how long it takes. We can understand and measure things. And as you've also said, we can start finding ways that allow us to understand that in humans, by having these biological markers, like looking at the urinary oxygenation or the fibrillary protein released by the brain. This is extraordinary stuff. It's called translation. We are translating what we understand in the large animal model and the depth of understanding that we can achieve there to what happens in human beings. And having the group of clinicians and first-class physiological researchers together means that we can study interventions, we can study diagnostics, and then we can take them to the clinic, we can take them to patients. We're number one in the world in this kind of process in diseases that matter to human beings and will matter probably to me pretty soon. And it matters very much to me that we get it right. And we can only do it here. And I hope you will continue to support us. And I hope the community understands how important this is. And I want to congratulate the young team of investigators for doing something absolutely extraordinary. Thank you very much for your attention.
I think you'd agree we've had an absolute tour de force tonight, and I think we should all absolutely congratulate each of our speakers. Before closing, I'd like to return to Lachlan's vignette, that is back to 1953. And I think we would all agree that John Gibbon was an absolute trailblazer and really was before his time in terms of no doubt bringing together a group of people with the concept of multidisciplinarity. That is the nexus between clinicians right through to engineers, I'm sure. And he would be chuffed, no doubt, today to see the same group. And I'm very much anear with Rinaldo's comment, sort of really chuffed to see young investigators rise to the challenge of solving wicked problems. And I think that it might not end on the first page of the New York Times, it might be on page 18 or whatever, but we can see that they're well on the road to profound changes for the benefit of us all, and for that they should be congratulated.